Cell is the basic structure of all living creatures. Today we are going to study about the cell and its basic structure. When we think about studying cells, few basic questions come to our mind. That are all the cells same? Is there something that makes the cell different? Is it one thing or several things? Is it their function that makes them different? Is it their location? Is it the organism, that is whether an animal cell is different from a plant cell, or a bacterial cell is different from plant cell or an animal cell. Does these things make the cell different from each other? What are the things that make them different? Their structure, their function, what else? And the third question that comes to our mind is, do these structures have the same function, like they function, all of the structures inside the cell, do they perform the same function, or they have different function, or they have their function supporting each other, or one is doing one thing or the other is doing a totally opposite thing. So we are before diving into the structure of cell, let's think about these structures and try to solve these questions today. Can you notice on these images, the first one is an animal cell, the second one is plant cell, and the third one is bacterial cell. These are different from each other, not in just in their structure, but within the cell, the organelles, the things present inside the cell are different. In the plant cell, the things are more green. There are some structures that are filled with green color that are absent from the animal cell, and there are other structures that are present in the animal cell that are absent from the plant cell. And then this one, the last one, the third one, is the bacterial cell, and you can see this is the flagella. The flagella is this is the flagella coming out from the bacterial cell, and this cell is more simpler than the other two complex cells. The small parts or contents in the cell are called organelles. In this image, you can see that there is a cell, a plant cell, and it is covered by or contained within a membrane, which is called a wall or a cell wall. Okay, and then there is the most prominent structure is nucleus, and here you can see a green structure, and that is the chloroplast, which gives green color to the plant and it makes food for it. We will study all of the organelles separately uh, soon. And then there is mitochondria, and mitochondria is very important for the cell respiration, the breathing of cell. And then there are um, endoplasmic reticulum and Golgi apparatus, and there are a lot of organelles that we are going to discuss very soon. Here you can see the cells of an onion under a light microscope. You can clearly see the nucleus and the well-formed structure of the boundaries of a cell. Now we are going to draw the cell by our. This is the structure of a cell. You can see that there is this cell membrane, and inside is a nucleus. You can see very clearly that this is the most prominent structure inside the cell, and the nucleus and the cell itself. The cell is filled with a watery liquid called the protoplasm. Protoplasm, and the protoplasm between the cell membrane and the nucleus is called cytoplasm, and the uh, protoplasm within the nucleus is called nucleoplasm. So cytoplasm is between the cell membrane and the nucleus, and protoplasm is inside the nucleus. nucleoplasm is inside the nucleus, and together cytoplasm and nucleoplasm, they are called, they together form the protoplasm. So there are various organelles present inside the protoplasm. Okay, you can see the nucleus here very clearly, and inside the nucleus there is DNA. DNA is actually, if we make it straight, it will be a 2 meter long, very long, very long. So inside the very small nucleus, it is in the form of coils, very, very convoluted coils, very spiral, too much spiral, okay, in the form of too much spirally DNA. And there are regions inside the nucleus, that is called nucleolus. Usually only one is present, but you may find more than one. What happens is that things that form the nucleolus, they come at certain time, they come together at certain time, they perform their function, and then they disassemble, this get separated, and the nucleolus finishes. So usually you will find only one nucleolus inside the nucleus. So the function of nucleolus is that it forms ribosomes using the information that is contained within its own DNA, and it also helps to form uh, nucleolus also contains the genes for making ribosomal RNA and transfer RNA. These are different kinds of RNA that are used, that to perform the function of making the proteins. Okay, and outside the cell, so outside the nucleus, there are various the nuclear membrane. This is the nucleus is covered by a membrane, which is again a double layer membrane, and it is called nuclear membrane or the nuclear envelope. There are several pores, small, very, very tiny pores. Pores, I mean by pores, I mean holes inside the nuclear membrane, and those are called nucleopore. And their function is to the transfer of or exchange of materials between the from cytoplasm to nucleoplasm. The things go from nuclear pore through the nuclear pore, from nucleoplasm to cytoplasm, and vice versa. Okay, and then we have several structures like mitochondria, Golgi apparatus, and endoplasmic reticulum, ribosome within the cytoplasm of the cell, means they are outside the nucleus. Inside the cytoplasm, uh, there are structures which can be seen through microscope, and these are very important structures, and this is called the mitochondrion. Mitochondrion is singular, like for one mitochondrion, and the mitochondria, you replace on with a. Mitochondria is the plural word for mitochondrion. So there are a lot of mitochondria present inside the cytoplasm, and they are very important for the function of the cell. The mitochondrion itself is like an oval shaped kind of a structure, and it has two membranes. The inner membrane is folded inside the mitochondrion, and the folds are called, or the folded form of this mitochondrion is called cristae. It's called cristae. And the fluid inside the mitochondrion, or you can say the cristae, is present inside uh, the fluid which is called matrix of the mitochondrion. Like mitochondrion has the matrix as its base, and cristae is the folded membrane, the folded inner membrane of the mitochondrion. So mitochondria are very small, about only 1 micrometer diameter, and the more the cell has to work and do aerobic respiration, it is very important for the aerobic respiration, means oxygen is required to perform this function of respiration. And by respiration, it produces more energy. The more energy a cell needs, the more mitochondria it has. The more you are going to use for exercise your muscles, the more mitochondria your muscles are going to produce. And liver cells, like for example, liver cells has a lot of mitochondria, around 2000 mitochondria, because it performs a lot of function and a lot of things, a lot of work is going on inside the liver. uh, how does it produce energy? It converts ATP. ATP you will uh, soon study it further, that ATP is the form which is converted to ADP, and hence it produces energy. And this process takes place, this is the process of aerobic respiration, during which ATP is converted into ATP and inside the mitochondrion. The most, it is like the breathing center for the cell. Okay. Okay, now Golgi apparatus are single membrane structures, and they are present inside the cytoplasm like this. They are actually stack of flattened sacs. Like there are sacs that are flattened and stacked together. Okay, this is they are not attached to the cell membrane. They are inside the cytoplasm, somewhere between the nucleus and the cell membrane. Okay, and there are continuously they are forming, and their small parts are separating from it, and this small part that are separating from it are called the vesicles. These are, like, I'm drawing it like these are the vesicles that are being separated from the main cistern. So these sacs, the sacs are called cistern, and the since it's continuously forming and separating, so the vesicles are produced, and these are called the Golgi vesicles. Okay, and the Golgi vesicles, they have function to carry their contents to other parts of the cell. And the Golgi apparatus, to the cistern and the vesicle together, they are called the Golgi apparatus. The Golgi apparatus functions together, the whole apparatus functions, uh, the function is to chemically modify the molecule it transports. For example, if it transports sugar, then it can add proteins to make it glycoprotein, and then transport it, means it is carrying sugar, and it is also transforming it from one place to another. It carries, the vesicles carry, later on carries it from one place within the cell to another, or carries it to the cell membrane otherwise. Okay, another